Hi, great. So thanks so much for the invitation to speak. I'm going to discuss our work on fast multimodal imaging of neural dynamics across sleep and wakefulness. And Ellen encouraged me to specifically focus on EEG fMRI, so I've dedicated a couple kind of sections to methodological issues in that space as well. Um, so sleep transforms nearly all aspects of brain physiology and brain function. As we fall asleep, there's this really profound transformation of, activity across really distributed brain networks, as well as a shift in behavior and the function really in a widespread sense of the body. And this process is really essential for brain function. So a single night of bad sleep will really typically feel the next day in terms of worse cognition and attention and mood. But disrupted sleep across the lifespan also has significant consequences for neurological health. And poor sleep in midlife, for example, has been linked to the onset of neurodegeneration decades later. So clearly during sleep, there's essential processes taking place that preserve the neurological and cognitive function of the brain. Despite the importance of sleep, it has classically been very challenging to study because it really poses a challenge for neuroimaging. Since activity is really changing throughout the whole brain simultaneously, and many different kind of aspects of how the brain functions, ideally to understand what's happening during sleep, we would use multimodal imaging methods that would allow us to both survey the whole brain at the same time, as well as really access these different types of things that are happening in the sleeping brain. And so I'll describe just a few of those here, just a small subset of the different functional effects that sleep has on the brain. The first is the one that we're really introspectively aware of, which is the major cognitive change that happens at sleep onset. So we shift from a really kind of alert, engaged mode where we're kind of interacting with our environment to a very inwardly focused state where external behavior is suppressed, and we either become unconscious or perhaps enter a vivid dreaming state. As this cognitive transformation takes place, we also know that there's very well characterized kind of stereotypic changes that we can measure in the EEG. This is an example just of the human EEG in the transition into non-REM sleep. And you can see that in wakefulness, the EEG has this kind of very low amplitude, desynchronized pattern. As people transition into sleep, you start to see these individual oscillations like sleep spindles and individual slow waves called K complexes. And by the time people reach the deepest stage of sleep, these slow waves start to become continuous. And you see a kind of constant, slow, low frequency dynamic in the neural activity. And so these EEG changes are actually viewed as the defining feature of sleep in the human. So if you want to tell what stage of sleep somebody's in, you would do this EEG, look for these patterns, and determine which sleep stage it is. And so many decades of work have focused on how the neural dynamics that these EEG patterns reflect might potentially support the cognitive effects of sleep. A much more recent area of interest for sleep has been the idea that sleep might also be important for basic functions like waste clearance from the brain. The study from the Nedergaard lab in 2013 measured amyloid in the mouse brain, and they found that mice that were asleep were able to remove amyloid from the brain at much faster rates than they were able to during wakefulness. So this brought about this idea that maybe the implication of sleep and neurodegeneration could be related to a potential role for sleep in sort of actively transporting solutes out of the brain. And following this work, several kind of subsequent studies have implicated sleep and sleep-wake cycles in the onset of neurodegenerative disorders and levels of amyloid and tau. So what is happening during sleep to potentially kind of contribute to these very different electrophysiological and kind of waste properties that have been reported in these different systems? Since it's many interconnected brain networks that are engaged during sleep, we'd ideally like to be able to study many different things about the function of them at the same time to potentially understand what's happening. So during sleep, as I mentioned, it's really characterized by changes in neural oscillations throughout the cortex, which we can measure with EEG, which are kind of indicated here in green. And extensive animal work has also shown that deep brain structures like the thalamus and the brainstem and the hypothalamus are key control structures that can actually switch animals between states of sleep and states of wakefulness. So ideally, if we want to understand circuits that are contributing to sleep, we would be able to image really throughout the whole brain and capture these small, deep brain regions. And finally, given that sleep has been implicated in these really basic metabolic functions, we'd also like to image not just what neurons are doing, but other potentially non-neuronal physiological changes that might play part of the role of what's happening during sleep. And so my lab focuses on how we could potentially address these questions in humans using multimodal neuroimaging. So we integrate EEG, which lets us look at sleep stages and different kind of neurophysiological dynamics that happen during sleep, with high spatiotemporal resolution fMRI. We use both fast fMRI to let us look at changes across these deep brain structures that are happening really on sub-second timescales, as well as taking advantage of ultra-high field fMRI to look, for example, into the thalamus as one of these kinds of much deeper, smaller regions. That's very important for sleep regulation. And finally, for using different non-neuronal contrast mechanisms in fMRI to potentially see what sleep is doing to other facets of the brain, like the flow of cerebrospinal fluid. And so today I'll talk about just kind of two projects within this space, focused on first understanding what are the consequences of sleep for the brain. In particular, we wanted to ask whether the flow of cerebrospinal fluid in the brain changes during sleep. And second, what are the potential control circuits for sleep? In particular, animal studies have shown that many specific brain regions can causally control sleep and wake. What's less clear is how these different brain regions work together. And so we wanted to be able to image thalamic circuits contributing to arousal regulation at transitions between sleep and wake states to understand how these are potentially evolving in the brain spontaneously. And so for both of these questions, we're going to use multimodal neuroimaging to simultaneously measure really distinct aspects of brain function. Okay, so in part one, we wanted to investigate the fluid dynamics of the sleeping brain. And so a kind of key motivation for this study is firstly, we know that sleep, it changes really dramatically across the lifespan and is very important for brain health. This is just an example of kind of typical sleep efficiency in aging with kind of years along the x-axis. And you can see that there's a strong evolution of sleep as we age. And as I mentioned before, this has been thought to potentially couple with the fact that sleep may be important for kind of actively transporting waste products out of the brain. And so given these observations, it's clear that sleep is really important for preserving neurological health across the lifespan. And we wanted to understand whether there's potentially specific fluid changes in the brain that might potentially be changing during sleep. And so we focused in particular on cerebrospinal fluid, CSF. So as you know, our brains are enveloped in CSF, which kind of cushions the brain, as well as forming a primary waste exit route for the brain. And so at slow time scales, there's kind of a classically known circulation for CSF. CSF is kind of constantly produced throughout the day and constantly drained. But what hadn't been investigated is whether the dynamics of this CSF flow might potentially change during sleep. And so that's what we wanted to design a study to answer, is asking whether the way in which this fluid moves during the brain might potentially be different during the sleeping state. Many decades of work had already investigated what governs CSF flow in the awake human brain, including sort of the original MR studies of this in the 80s. And they found that a primary driver of CSF flow is respiration, as well as breath holds. So for example, in this study, the black bars here are showing paced breathing, and you can see that there's strong kind of CSF flow coupled to those breaths. And in addition to that, the cardiac cycle is also a kind of typical driver of CSF in the awake state. So when we're awake, CSF is kind of always pulsing with every breath and every heartbeat. And very high-resolution imaging studies in mice have also kind of examined the fluid flow along vessels themselves. What they've observed is that with each kind of pulsation of the vessel, which happens, for example, with every heartbeat, you actually get motion of the fluid and of any particles contained within the fluid along the vessels. So an increasing idea has been that potentially changes in these vascular systems might actually be able to kind of contribute to the flow of cerebrospinal fluid in the brain. What's less clear is why or how that might potentially change during sleep. So we conducted a study using simultaneous EEG and fast fMRI to look at this question in the sleeping human brain. We wanted EEG to look at whether they were asleep or not, as well as looking at the specific neural dynamics happening during sleep. We used whole brain bold fMRI to assess hemodynamics throughout the brain, or that is most of the brain losing the tip of the temporal lobe during sleep. And we also designed an approach to use this fast fMRI to measure the flow of CSF, which I'll explain more on the next slide. And we wanted to scan subjects who were just spontaneously sleeping in the scanner. So we asked our participants to restrict their sleep to just 4 hours the night before the scan. They would come in around midnight, and then we would scan them with this simultaneous EEG and fMRI approach. And when I say fast fMRI, in this case, we were using a TR of 367 milliseconds, giving us kind of sub-second tracking of hemodynamics throughout the brain. So this yellow box here shows the typical positioning of our acquisition volume. And you can see that the bottom slice here is intersecting with the fourth ventricle. And so the key way for us to image CSF flow was to focus on positioning this such that the bottom slice intersects with that ventricle. If I blow up a slice here, you can see that there's actually a very bright signal coming in at this point from the ventricle, which is indicating upwards flow of CSF. So the technique that we took advantage of in order to be able to use fast fMRI to measure CSF flow is turning what's classically been an artifact in fMRI into, in this case, our signal. So fast fMRI is vulnerable to this artifact called flow-related enhancement or inflow effects, which is essentially that as you scan very rapidly, everything you're imaging reaches a kind of steady state with a kind of dimmer image intensity than it would have initially. But if a new fluid arrives into the imaging volume, which hasn't previously been imaged, it has much brighter signal intensity. And so you can get a bright signal that corresponds to the velocity of that incoming fluid. Specifically, if fluid is flowing up relatively slowly, we might see bright signal in the bottom slice, which then kind of disappears in the upper slices. If fluid is flowing so rapidly that it exceeds the critical velocity of our imaging, you can actually then see that bright signal along several of the bottom slices. So by taking advantage of this approach, we were actually able to simultaneously measure the hemodynamics as well as the upwards flow of CSF through the ventricle in the brain. As we do this, this is an example of one person who we scanned while they were sleeping in the scanner. We're able to simultaneously monitor what type of brain state people are in. So this is a person here. This top panel is showing the EEG spectrogram of this person while they're sleeping. On the left part in blue here, this person was in non-REM sleep, specifically mostly stage N2. Here in this kind of bright part, this person is waking up. We see some artifacts in the EEG at this point linked to kind of the motion that they have while they're awakening. And then they reach a kind of stable awake state for the rest of the scan. You can see this red stripe at 10 hertz, which is the classic occipital alpha rhythm, which is a sort of classic indicator of wakefulness. And in about half of our subjects, we actually also asked them to do a spontaneous self-paced task just to confirm, behaviorally whether this corresponded to sleep. And indeed, for example, for this person, you can see that they're not performing the task during the sleep period. They kind of remember where they are here when they wake up, and then they kind of continuously perform the task after that point as they remain awake. And so finally, I'm showing in this case the CSF signal from just even a single voxel. And what we saw was this really dramatic change in CSF flow, specifically when people were asleep. You can see when this person was asleep, there are these huge slow waves of CSF flow that are appearing with this kind of very large amplitude and relatively slow timescale. As soon as the person wakes up, these large waves disappeared. And then when the person was awake, they were replaced with this much faster kind of continuous pulsation with the breath cycle, which is what people have typically seen during wakefulness. So from the single data set, we could already see by eye, there's this really dramatic change in CSF flow during sleep in that it starts to kind of flow in these high velocity, large waves upwards into the brain during sleep. This is an example from one participant. But if we look across all of our participants, we really kind of consistently saw this effect. So this power spectrum here on the left is just the power of the CSF signal in all of our subjects in sleep versus in wakefulness. And you can see in blue here that during sleep, there was a strong increase in the spectrum peaking at around 0.05 hertz. And so typically these waves would occur at a rate of approximately every 20 seconds or so during the sleep state. And we saw this dynamic really consistently that when people were asleep, we would get this kind of increased pulsation of CSF flow. So where might these CSF waves be coming from? So as I mentioned before, one of the key ideas for what might potentially be contributing to CSF flow is the idea that potentially pulsations actually in the blood vessels themselves could potentially drive this. And we had acquired a hemodynamic signal because we were performing bold imaging at the same time. So we next looked at how the bold and the CSF signals co-varied during sleep. The bold here is in green and the CSF is in purple. And what you can see is that every time we saw a wave upwards of CSF flow, this corresponded to when we had a kind of profound dip in the bold signal. So whenever the bold signal would decline, that's when the CSF flow would rush upwards into the head. The bold signal, of course, does not directly measure cerebral blood flow or cerebral blood volume, but what we notice is that the CSF signal seems to be most tightly coupled specifically to the temporal derivative of that bold signal. Specifically, when bold is dropping is when CSF flow appears. So this would be really consistent with an idea where potentially when blood flows into the brain, to preserve the kind of intracranial volume within the skull, CSF must flow out. Whereas when blood flows up, when blood volume decreases, then CSF actually has an opportunity to flow in and kind of fill the space that's left behind that blood volume. So we found this was really consistent with a model of large waves of blood volume versus CSF alternating in the brain during sleep at this rate of approximately every 20 seconds or so. So this is an example of what this looks like. This is data from a single subject sleeping in the scanner. In this video, the red is going to be the blood oxygenation level dependent signal in this person. Because they're sleeping, you're going to see it's changing in these large slow waves. And then here at the ventricle, we're able to measure in blue the upwards flow of CSF. And what we saw is that what you'll see in this video is that as each kind of red wave dissipates, that's when you get this upwards flow of CSF into the brain. So here we go. There would be this large red wave, and then the flow of CSF, and then another bold wave, and another flow of CSF. And so these two kind of continuously would alternate with each other, throughout most of the sleeping period. Okay, so then we became very intrigued at what might be happening that could potentially be contributing to these really large scale flow waves that are specifically happening during sleep. What might be changing during sleep that could actually contribute to seeing these huge waves of bold, which are much larger than we would typically see during the awake state. So we looked at what was happening around each individual CSF wave. So time zero here is the peak of the flow wave of the CSF. And what we found is if you look at the temporal derivative of the bold signal, that dips just prior to that. So this is really consistent with this alternation of the blood and the CSF. But about 6 seconds earlier, we found this rise in the slow delta power of the EEG. So these slow waves are this kind of classic signature of non-REM sleep. And we know that in the human brain, when you see a kind of slow wave on the EEG, what that represents is the suppression of neuronal activity throughout a large expanse of cortex for many hundreds of milliseconds. So what this essentially represents is first the neurons kind of go quiet in this EEG slow wave, then we get a dip in the bold signal, and finally we get this upwards rush of CSF into the brain. So this was very intriguing to us because it suggests one potential contributing mechanism to how CSF flow might be shaped within the brain. It could be specifically that, as neurons change their activity in very low frequency synchronous patterns, because of neurovascular coupling, they actually have a mechanism to kind of modulate the flow of the brain, of the CSF in the brain. And to look at this a bit more closely, we also deconvolved what would be the impulse response if we tried to predict CSF flow just using our EEG slow delta power. And what we found is that if you convolve the EEG power with the best fit impulse response to predict the CSF, first, we're actually able to predict CSF flow using just the EEG alone. And second, this coupling factor is this kind of slowly evolving smooth response, which actually looks somewhat similar to the classic hemodynamic response function. Again, very consistent with a vascular mechanism that's driving CSF flow during sleep. It's important to note that we don't have kind of causal evidence in this study. Many different things change in the brain and body at the same time when a slow wave occurs. So I think one contributing factor is local neurovascular coupling to slow waves. When neurons go quiet, you would get a drop in blood volume that could contribute to this flow. A second key mechanism is that we know from high resolution animal studies that arterial or vasomotion, which is this kind of oscillatory intrinsic dilation and constriction of the arterial wall, is also entrained by high frequency neural activity. And so a slow envelope in the neuronal activity can actually entrain blood volume changes, providing another kind of way to get this vascular change during sleep. And we also know from Osbe et al. Study that when you see K-complex in the neural activity during N2 sleep, there's also a very systemic vasoconstriction, which is also even present in the fingertip. So there also seems to be very widespread vascular changes coupled to autonomic state changes during sleep that are likely also contributing to this phenomenon. So altogether, this kind of suggests a model where during sleep, when we start to have these individual slow wave events, they're able to be coupled to large scale changes in the vasculature and blood volume of the brain, which in turn, through kind of mechanical volume displacement, can actually induce these very large scale changes in CSF flow. So to conclude just this first part, we found that there's very large waves of CSF flow that appear during non-REM sleep. Most of our data was from N2 sleep, and so that seems to, at the very least, be a state where there's quite a bit of this flow. And by using the simultaneous EEG and fMRI, we were able to find a coupling between neural activity and the CSF flow, which was very consistent with a model where one kind of driver of CSF flow is that neural activity can actually induce CSF flow because of its effects on the vasculature. And I think really importantly, this study does not actually measure waste clearance. We're just measuring very large scale bulk flow in the CSF. We were actually quite surprised to see such striking changes in this very macroscopic flow. And so I think future studies will really be needed to understand what are the consequences of large scale CSF flow in the ventricle for waste clearance, which ultimately actually needs to really happen first in the tissue and then ultimately exit through these fluid systems of the brain. So I'll move on to the second part, where we wanted to examine thalamocortical dynamics at the moment of awakening from sleep. And so extensive animal studies have shown that there's dozens of brain regions that change their activity at sleep-wake transitions. Many of these regions, if you kind of go in and stimulate, you can also induce these sleep-wake transitions. So there seems to be, a very distributed network for potentially modulating arousal state transitions in the brain. What's much less clear is what dynamics across these networks actually happen spontaneously at these transitions. Animal studies have classically found it challenging to record from many of these regions at the same time because it requires kind of inserting electrodes targeting each region. And so fMRI is actually very well suited to asking this type of question since it provides that sort of 3D whole brain imaging. So we wanted to go in and ask a very kind of simple question about these brain circuits. If we look at a structure that's been really implicated in arousal regulation, and we divide it into all of its different nuclei, is it really the case that this brain structure is just all kind of activating when we wake up from sleep? Or could it potentially be the case that actually there may be very distinct dynamics in the different nuclei of that brain structure that we might potentially be able to track using very fast imaging to kind of try to understand how these dynamics are evolving over time? And so we focused in particular on the thalamus. The thalamus, of course, is this sort of central deep brain structure, which is very intricately interconnected with the cortex in these kind of dense feedback thalamocortical loops. And extensive animal studies have shown that if you stimulate the thalamus, you can drive arousal and awakening from sleep. By stimulating, for example, like one region or just a collection of regions, you can actually kind of change the arousal state in the cortex as well as change the behavior of the animal. What's less well understood is that, thalamus is really not one brain structure. It really contains, at least a dozen very distinct nuclei with really different functional and anatomical properties. And what's less clear is whether all of these nuclei are kind of acting in concert and doing the same thing, or whether there's potentially different functional roles for these different subnuclei of the thalamus, which would really require the ability to image them simultaneously to understand if they have similar or separate roles at state transitions in sleep. So our goal was to understand if we can see different timing of neural activity across the thalamic nuclei at the moment of state transitions. And so prior fMRI imaging studies have already kind of beautifully shown that at an arousal event from sleep, there's a strong activation of the thalamus. So thalamus is more active when you wake up than it is during sleep. Our key goal now was to look at these really diverse, distinct nuclei of the thalamus and understand exactly when do they activate and are they all behaving similarly as we undergo a state transition. So this study was led by Beverly Setzer, a really talented PhD student in the lab. So she wanted to ask, do these different nuclei potentially have different roles in awakening? And so to do that, we then turned to 7T fMRI. The human thalamus is relatively large, but to subdivide it into these much smaller individual nuclei, we really needed the higher SNR that's provided by ultra-high magnetic field, by ultra-high field MRI imaging. And so we moved to doing this at 7T. And again, we're using a very fast TR here, 247 milliseconds, to potentially look at the dynamics of each of these regions over time. And again, we're also scanning at midnight so that we can catch people who are just undergoing spontaneous state transitions as they're sleeping inside of the scanner. And we focused on awakening because although the transition into sleep can be quite gradual and fairly subtle over time, often awakening really happens at a specific moment, which is kind of a more abrupt transition, which would allow us to look at relative timing locked to that event. And so in this study, we ended up defining the moment of arousal transition as the resumption of behavior. We asked people to press a button when they were awake, and then the moment where people were able to recover performing that button press is defined as the kind of switch in behavioral arousal state that we saw. So most of the study really focused on behavioral arousal, but in a subset of subjects, we wanted to perform simultaneous EEG fMRI, again, because EEG would sort of confirm to us that our behavioral task definition really does represent sleep. And so there's been really kind of at least a decade and a half of very great work on this extremely challenging problem of how to perform simultaneous EEG fMRI at 17. So both the image and the EEG artifacts become really challenging at ultra high field. The cap can produce kind of image signal loss as well as the field itself, really kind of creating very strong artifacts in the EEG, which in my view are really the primary challenge for performing this at 17. So to do this, we've been collaborating with Georgia Bonmasar at MGH, who's designed this very cool EEG cap that replaces the copper wires with printed conductive inks, which gives really beautiful image quality and sort of resolves the first part of this challenge. The second, more challenging part is how to actually get high quality EEG signals. And so we've been using the method originally proposed by Luo et al. And Chowdhury et al. In 2014, which is to isolate a subset of the EEG electrodes from the head while still recording EEG from the others. So essentially, some of your electrodes just record noise without recording the signal. And this objective measure of the noise really facilitates the ultimate artifact removal from the EEG. So we've taken this method and updated it with new kind of dynamic signal processing methods that allow us to kind of very dynamically model and subtract the noise signals recorded on these kinds of reference channels from the EEG signals. And using this new package, we've been able in a subset of these subjects to actually acquire usable EEG data at 7T across these spontaneous transitions between sleep and wakefulness. To really validate this approach, we did want to start in the visual system so that we could provide a very controlled input and make sure that we're getting the signal that we expect out. This kind of just gives us a sense of, is this artifact removal method working as intended, and are we in fact able to recover good EEG fMRI correlations? And so here's an example where we used a visual stimulus with a 12 hertz flicker. So we're looking for a 12 hertz signal inside of our EEG. You can see that in the raw data, we're really not able to recover that signal. And a classic method like OBS, which is what's implemented in many existing software packages, also cannot recover that signal. But when you use this reference-based dynamic approach, we now get this huge reduction in power, and you can finally see this 12 hertz peak pop out of the power spectrum, indicating that we've successfully recovered our 12 hertz stimulus-induced signal. And what I thought was really beautiful in these validation data is that then if you kind of run a correlation of just EEG power with your fMRI signal, you get the beautiful expected correlation in the visual cortex, but you also get this really nice signal pop out in the LGN. Which just goes to show that, you know, in this single 4-minute run, the 7T fMRI data gives us such high quality imaging of these tiny thalamic nuclei. Okay, so using this, we next wanted to go and examine the moment of these behavioral state transitions at awakening. So in the subset of subjects who had EEG, we were able to look at how do their EEG dynamics change at the behavioral state transition. Here's an example of someone who starts performing the task. When the black circles appear, they've done their first button press. And you can see from this arousal EEG spectrogram that when they kind of do the button press corresponds to this state transition in the EEG. They go from this N2 sleep with spindles to abruptly having those disappear, and suddenly this 10 hertz stripe comes on, showing us that this person is awake. If we looked across arousals, we also kind of consistently saw this rise in alpha power that was locked to the behavioral state change that we were measuring. And so next we wanted to go look at what the thalamus was doing. And so we did indeed see there's this really strong activity in the thalamus in red at awakening as compared to the cortex in blue, which is very consistent with prior studies. But what we realized is that in fact, this rise in thalamic activity is starting at least 3 seconds or so before the person actually executes the behavioral state change. So first thalamic activity starts to rise, then you get the awakening, and then subsequently you have this large scale suppression suppression of the fMRI signal throughout the cortex. And this is very coarse for me to just kind of show you all of cortex. If we break it down into its individual regions, you can see that a couple cortical regions do have this more thalamic-like signal of activating before arousal. Specifically, the anterior and posterior cingulate kind of popped out as more thalamic-like dynamics with this strong activation at state transitions. If we take a more data-driven approach and just kind of put in the signals from all of our different thalamic and cortical regions, we can see that nearly all the variance is explained by just the first two principal components. Again, this purple one is kind of popping out as this strong increase in activity before arousal. And then you can see from these bright red colors here, the thalamic nuclei really have the heaviest weighting, contributing to this kind of arousal-coupled increase, whereas most of the cortical regions are instead weighted on this first principal component, which shows this widespread decrease. So next, we wanted to say, okay, well, if the whole thalamus is activating, still, are each of these nuclei really doing the same thing, or do they potentially have different temporal dynamics? And so what I'm showing here is this kind of slow rise in activity across just a subset of our thalamic nuclei. And you can see that all of them are increasing before the arousal at time 0. But some of them, like centromedian thalamus, are increasing incredibly early, more than 8 seconds before the behavioral state transition. Whereas others, like the pulvinar and the ventroanteral nucleus, are increasing much later in time. So this spread was on the order of several seconds. And if we kind of tiled it across all of the thalamic nuclei, we saw a very consistent temporal sequence with the centromedian and ventroposterior lateral thalamus consistently activating early, and then a kind of subsequent evolution across the rest of the thalamus multiple seconds later. So by scanning very rapidly, we were essentially able to uncover that while all of thalamus does activate at arousal, it seems to really originate in a couple core nuclei, only later propagating to the rest of the nuclei and ultimately resulting in this behavioral state transition. A really important thing to keep in mind with fMRI, of course, is that we're measuring a hemodynamic signal. And so you can also get very different hemodynamic response properties across brain regions, potentially leading to earlier signals that just reflect the vasculature in a given brain region is really fast. So we also conducted a separate experiment using a breath hold to control for just intrinsic differences in cerebrovascular reactivity across these regions. And we found in this kind of hemodynamic lag analysis that overall the thalamic nuclei are very similar to each other in terms of the vascular responses, with the exception of the interoventral nucleus, which actually was a bit late. Maybe there's sort of a large vein there or some other kind of feature of the vascular anatomy that makes it late. But overall, it was clear that these cerebrovascular reactivity differences could not explain this very consistent temporal sequence that we were getting locked to arousal, leading us to think that the most likely origin of this is really a kind of neural change, which is reflected first in the CM and BPL and only later in the other thalamic nuclei. Finally, we had the opportunity to ask whether this sort of temporal sequence really reflects just the moment of transition between arousal states or the fact that people actually subsequently remain awake and there is an overall state difference. And so what was interesting is that we actually had quite a diversity in our data of people who either, pressed the button and then went right back to sleep, or people who pressed the button and then really managed to sustain this higher arousal state and continuously perform the task for several more minutes. And so we divided all of these data into these sustained in pink or transient in purple arousals, and recalculated what was happening in the thalamus. And what we found is that in both cases, you actually get this very striking increase in thalamic activity. So even just a very brief transient arousal still has this large activation that begins before the moment of the behavioral change. But people who successfully then maintain the higher level of arousal have this kind of sustained signal in the thalamus, whereas people who lapsed back into a low arousal state would have a kind of immediate dip in the thalamus and it would return to a lower baseline. And interestingly, we didn't see nearly as striking of an effect in the overall cortex. If we broke it down by cortical regions, there are a couple that are showing some more subtle differences between the sustained and transient cases. What I think is kind of really cool is how this potentially aligns with evidence from other labs using electrical stimulation. So for example, this recent paper from Bastos et al., they stimulated the intralaminar thalamic nuclei using electrical stimulation. And so one of the targeted regions is actually the CM, which is one of the regions we found to be quite early. And they found that even in an anesthetized animal, stimulating the thalamic nucleus can actually cause them to awaken from anesthesia, even while still breathing, even while still being under the influence of the anesthetic itself. So there does seem to be a kind of strong causal effect of CM on arousal, both in terms of behavioral and electrophysiological signatures. So I would really love to see kind of these other nuclei that we identified as being on the earlier side to potentially be investigated in future animal studies to see to what degree do these temporal properties relate to the kind of causal consequences you can probe in the model system. All right, so to wrap up this second part, we found that cortical and thalamic activity overall diverge at state transitions, with the cingulate being an exception that kind of looks more thalamic-like. But using this very fast imaging, we were also able to identify a specific temporal sequence of thalamic activity that precedes transitions in arousal state. And finally, I think this is also just really exciting because it shows that fast 7T fMRI has the ability to really resolve and distinguish sub-second dynamics across different thalamic nuclei. And there's a huge range of interesting cognitive neuroscience questions that could potentially benefit from this approach to look at sequential timing across these deep brain regions, which are so important for many diverse aspects of cognition. So finally, just to kind of wrap it all up, I showed you guys that neural activity and CSF flow are coupled during sleep, which has suggested potential mechanistic pathways regulating CSF. I also showed you that the transition to awakening is preceded by activity in a very specific constellation of thalamic nuclei. And I think finally, these studies just kind of highlight the rich information present in multimodal neuroimaging data, particularly as we push to higher spatial and temporal resolution. There's a huge amount of information embedded. And in our studies, we really haven't taken full advantage yet of the coupling of these EEG and fMRI signals. And so I think that's going to be a really rich area moving forward. And finally, I'd like to thank my fantastic lab members at BU, especially Stephanie Williams, Beverly Setzer, and Nina Foltz, who've been contributing to this work, as well as my great collaborators at MGH, especially John Palameni and Georgia Bonmasar, who were collaborators on these studies, and the generous support from our funding sources. I look forward to answering any questions you might have.